Hello and welcome to the Enzymes revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Enzymes are protein molecules that act as biological catalysts, so they speed up chemical reactions. Each enzyme will catalyze a specific reaction. The working part of an enzyme is its active site. The shape of the active site is complementary to that of the substrate. The substrate is the biological name for what we would call the reactant. So only this substrate is able to enter and bind to the active site. The enzyme amylase will catalyze the breakdown of starch into sugars. Other substrates such as protein or lipids will not fit into the active site. Starch has a hexagon structure and can bind to the active site of the enzyme, whilst the protein has a circular shape and it will not fit into the hexagon active site. The model that is used to represent enzyme action is known as the lock and key model. In this model, the active site of the enzyme is a lock and the substrate is a key. Each enzyme has an active site, which the substrate will enter and bind to. An enzyme-substrate complex is then formed. A chemical reaction occurs, resulting in the formation of a product, which leaves the active site of the enzyme. Factors that affect enzymes. Enzymes are affected by changes to temperature and pH. From A to B, as the temperature increases, the rate of the enzyme reaction will increase. At higher temperatures, the molecules have more kinetic energy, so they move faster and there are more collisions per unit time. It is more likely that an enzyme-substrate complex will form, so the rate of reaction will increase. At B, the maximum rate occurs, and this point is known as the optimum temperature. So it's the temperature which has the highest rate of reaction. Different enzymes have different optimum temperatures. From B to C, as the temperature increases, the rate of reaction will decrease. This is because the enzyme has become denatured. The high temperatures change the shape of the active site, and the substrate cannot enter the active site, so the reaction cannot be catalyzed. pH in enzymes. pH is a measure of how acidic or alkaline a solution is. Each enzyme has an optimum pH. This is where it has the maximum rate of reaction. Below or above the optimum pH, the rate of enzyme action will decrease. This is because changing the pH will change the shape of the active site, so the substrate can no longer enter and bind to the active site. At extremes of pH, the enzyme is denatured. Metabolism is the sum of all the chemical reactions in a cell or body of an organism. Enzymes are biological catalysts that can increase the speed of these reactions. In an organism, metabolic reactions need to happen quickly enough to sustain life. Enzymes are essential to speed up metabolic reactions, so they occur quickly enough to sustain life. Without enzymes, reactions would be too slow and the organism would die. Enzymes speed up two main types of metabolic reactions: anabolism, where small molecules are combined to form a larger molecule, and catabolism, which is the breaking down of a large molecule into smaller ones. Examples of anabolism include photosynthesis, where carbon dioxide and water are combined to form glucose and oxygen, or protein synthesis, where amino acids are joined together to form proteins. Examples of catabolism is digestion, where large insoluble molecules are broken down into smaller soluble molecules, such as starch, into sugars, or respiration, where glucose and oxygen react to form carbon dioxide and water, releasing energy. Thank you for watching. See our revision notes, the link is in the description.